All right, everybody. Dr. Zebrafish here from Vet Triage. We are continuing through JAVMA, January 15, 2015, and we are talking about pathology in practice. And there is a zebrafish. That's what that is. So let's get into it. This study comes from the Zebrafish International Resource Center at the University of Oregon in Oregon, as well as the University of Arkansas in Arkansas. Transgenic zebrafish with an AB background were imported into the quarantine room at the Zebrafish International Resource Center, which is a repository and distribution center for thousands of mutant, transgenic, and wild-type zebrafish. Their scientific name is Danio rerio, and they're used by the research community. Clinical and gross findings: the 12 imported zebrafish carried a transgene encoding CRE, recombinase, expressed in pancreatic beta cells on the AB background. The CRE recombinase transgene alone does not result in morphological phenotype. This line is typically crossed to another transgenic line containing LOXP sites to induce CRE-mediated recombination and expression of the LOXP flanked target sequence in pancreatic beta cells in the progeny. The fish were 233 days old. Histopathologic findings in sentinel fish included infections with Pseudoloma neurophilia, which is microsporidiosis, and gill epithelial hyperplasia and hypertrophy. Histologic and laboratory findings within affected regions of the intestines: the mucosal epithelium was hyperplastic, irregular folds, fusion of rugae with epithelial dysplasia. Cells displayed intense nuclear basophilia, marginated chromatin, prominent nucleoli, nuclear molding, and karyomegaly. Dysplastic epithelial cells had mild anisocytosis and increased mitosis with aberrant sunburst-like or rarely tripolar configurations. Morphologic features were consistent with Pseudocapillaria tomentosa, which is an infection. This is the organism here. This is there. Here. It has these two ends to it, I guess. These two ends make it characteristic of this parasite, including an alternating amphophilic to basophilic periesophageal schizosome in females, hypodermal bacillary bands, and rarely in gravid females, intrauterine larvated eggs that were barrel or lemon-shaped with symmetric bipolar plugs. That's what those are, bipolar plugs right there. Eggs were approximately 40 to 60 micrometers in length. In some sections of the intestine, capillarid eggs were free within the lumen. Numerous intraproprial venules were ectactic with attenuated endothelium, and individual venules were surrounded by perivenular cuffs of chronic inflammatory cell aggregates and extravasated proteinaceous fluid. In some venules, there was pavementing of neutrophils, lymphocytes along the endothelium. Neural microsporidiosis and gill epithelial hyperplasia and hypertrophy were observed in the fish examined. So here is the biopsy. So you can see there's longitudinal and cross sections of these parasites. So this says here, chronic ongoing proliferative enteritis and intralesional subepithelial Pseudocapillaria tomentosa nematodes, which are worms, in transverse and partial longitudinal sections. And the bottom is an example of the egg. There you can see it. They found in the tanks of these fish. So the diagnosis was Pseudocapillaria tomentosa infection in zebrafish. Comments: although some capillarids may go through an intermediate host before infecting fish, the Pseudocapillaria tomentosa can be directly transmitted between zebrafish. Clinical disease and death in the AB fish occurred as early as 2 weeks after exposure. The gill lesions likely contributed to behavioral changes, including rapid opercular beating, thereby increasing respiratory effort and ventilation and moving toward the water surface, called piping, in proximity to water inflow. No changes in renal hematopoietic tissue was observed, indicating that this was not the result of anemia. Microsporidiosis has been associated with underweight fish. It is typically a chronic progression and does not result in acute death. The AP outcross fish had no underlying disease. The possibility of early horizontal transmission of the parasite to the progeny should be considered. Pseudocapillaria tomentosa eggs could potentially be shed with gametes, and the effect of bleach solutions, typically used to surface sanitize zebrafish embryos, on the Pseudocapillaria tomentosa eggs is not known. The sugar flotation and double centrifugation of tank debris is an attractive non-lethal alternative to histologic evaluation for diagnosis of capillariasis. It can be used for routine and repeated monitoring for whole tank populations. In this pathology service for zebrafish, they've diagnosed this infection in zebrafish from research facilities in the United States, Europe, Australia, and Japan. The sharing of zebrafish lines between facilities, the lack of quarantine for transferred fish, the direct life cycle of the parasite, and the potential for nematode eggs to survive surface sanitizing procedures may contribute to its spread. It causes emaciation, hyperpigmentation, and death. Histologic lesions with chronic enteritis or intestinal neoplasms associated with Pseudocapillaria tomentosa has been detected in wide range of hosts, including 25 cyprinids, European eels, cod, salmon, and catfish. Among popular aquarium fishes, the nematode has been recovered from the intestines of goldfish, of guppies as experimental hosts, and tiger barbs, most likely resulting in death of captive tiger barbs. Conspecific larvae were also associated in the liver of non-tetra, noga lisfenbendazole and levomizole as potential treatments for non-encysted nematode infections in fish. In their experience for this lab, this infection is frequently found within tissues of the intestines of infected zebrafish. Recent publications describe anamectin benzoate or ivermectin and fenbendazole treatments for capillariasis in zebrafish. Well, there you go. All that stuff is news to me, and take it for what you will. I guess for veterinarians, it's just a matter of, I don't know, I guess those who work in the laboratories would find this type of paper important, and for those who own fish, keep in mind that nematodes are a problem with them as well. Appropriate quarantine and separation of fish, as well as adequate husbandry, is going to be key. But that's pretty much common sense for any of these animals that live in tanks like this in closed environments. So, all right, we'll see you again tomorrow. Bye-bye.